of comparative vertebrate embryology. Number of hours 2. Key issues, topic, plan. First, elements of comparative vertebrate embryology. Second, morphogenetic processes. Third, direct and indirect development. Fourth, metamorphosis. Fifth, secondary determination of sex.
In this lecture, we'll do a brief overview of animal reproduction and development. Some of this will be review from previous units. Earlier in the year, we talked about the cell division process of meiosis, that is a reduction division. It cuts the chromosome number in half, and this can be used to produce haploid gametes. This occurs in the gonads of humans or in the ovaries of females and testes of males. Fertilization may occur internally in the female reproductive tract or externally for animals that live in aquatic environments. Fertilization is going to bring egg and sperm, or two gametes, together to produce a diploid zygote. Animals may develop externally or internally. Animals that develop externally rely on a yolk as a food supply throughout their entire development. Animals that develop internally, like placental mammals, like humans, initially rely on a yolk for food, but they receive nutrients from the mother after that. So the yolk supply for animals of internally developing species is much smaller than the yolk of externally developing species. The female reproductive cycle prepares the uterus for potential pregnancy every 28 days. This is regulated by hormones that come from the hypothalamus and the anterior pituitary gland. These are tropic hormones that regulate the production of estrogen and progesterone from the ovary. In addition to these tropic hormones, FSH and LH, regulating the production of estrogen and progesterone, they also regulate what is happening in the ovary as one follicle matures each cycle and then ruptures to release the egg for possible fertilization. With estrogen and progesterone being released from the ovary, this is regulating what's happening to the lining of the uterus. So at the very beginning of the cycle, the lining of the uterus is shed, and then as estrogen increases, the lining of the uterus increases. Increasing levels of progesterone is also going to increase the lining and then maintain the lining of the uterus to support pregnancy. I don't expect you to know the specific names of LH and FSH, just understand that tropic hormones are being released from the anterior pituitary gland to regulate hormone production in the ovaries. A zygote, which is the first cell produced by fertilization, divides by mitosis in a process of cleavage, which is really just mitosis. We're just skipping some of the growth phases of the cell cycle here. So it's a more rapid form of mitosis because growth phases, G1 and G2 of interphase, are being skipped. So initially, the cells are getting smaller and smaller and smaller. The cells are organized into a hollow ball of cells that goes through the process of gastrulation. Gastrulation is going to start to reorganize the cells into layers. So when gastrulation occurs, one side of the ball of cells folds inward, and so you get an outer layer and an inner layer, and then some more cells reorganize themselves so you get a middle layer as well. Each layer is destined to become a different collection of cells. So this is the beginning of those processes of determination and differentiation. So here we see the single diploid cell, the zygote, start dividing by mitosis, and you can see that the cells are getting smaller and smaller because we're skipping those growth phases of the cell cycle. The cells start out as one solid ball of cells and then become a hollow ball. So there's this space in the middle. The cells are around the outside, and then gastrulation occurs when one side of that ball of cells folds inward, and you wind up with layers of cells. Here we're identifying those layers of cells. The inner layer is the endoderm, middle layer as the mesoderm, outer layer as the ectoderm. This chart here highlights some of the structures that develop from each of these layers, and you do need to know some of them. I don't expect you to know the whole chart. But the ectoderm, ecto-outer, this will become the skin and the nervous system. The mesoderm, meso-middle, will become structures involving the skeletal system, the muscular system. This is really everything in between the outermost layer and the innermost layer. And then lastly, the innermost layer, the endoderm, becomes the inner lining of your systems like your digestive system. So this is the layer of cells that the food touches, or the respiratory system, the layer of cells that the air touches, so on and so forth. There are some variations in the patterns of reproduction in certain species of animals. One variation from the pattern that we see in most animal species is hermaphroditism. Some species are able to exhibit this condition where the animal has both male and female gonads. Some are capable of self-fertilization, but most still need to find a mate because this is going to help to increase variation in the population. It's really not beneficial to the population for hermaphroditic organisms to carry out self-fertilization. Hermaphroditism can be an adaptation for sessile or stationary organisms, where they're less likely to find a mate of the opposite sex. So if all organisms have both sets of gonads, it's easier to find a mate that is capable of reproducing with you. Some animals exhibit sex reversal under certain conditions, where males can become females or vice versa. Sometimes it's based on environmental conditions. Other times it's based on the conditions of the population, whatever sex is needed. So coral reef fish, oysters, they can exhibit this change. So they're considered hermaphroditic, but they're not exhibiting both sets of gonads at the same time. Another variation in patterns of reproduction is sex determination in some reptiles is not dependent on the type of chromosomes that are inherited, but rather the temperature of egg incubation. And different species will show different patterns. A lot of times it's related to the rate of increasing enzyme activity, so increased temperatures, increased enzyme activity, and that can affect certain hormone levels, which will affect what sex the individual is. So this graph here is showing the sex ratio, so it's specifically tracking the percent male in relation to temperature. So you can see that at certain temperatures, there are no males being produced. They're all female. So these really low levels would indicate high levels of females in the offspring in that brood. Lastly, the last pattern that I want to talk about is parthenogenesis. Parthenogenesis is a method of asexual reproduction where unfertilized eggs develop into an adult. So this still involves meiosis and the production of gametes, but some gametes are fertilized and others remain unfertilized but still develop into an adult. This may be triggered by reproductive isolation, so that the population can continue on without having to find a mate, or it could be due to hormone fluctuations. It also is a way to quickly populate the habitat in an absence of mates. So it gets rid of that stress of needing to find a mate. In certain species of bees, males are the result of parthenogenesis, where egg cells develop through parthenogenesis into a haploid organism.
Thank you for your attention. Send your homework to email.